So our next speaker is Daniela Römer from the University of Würzburg in Germany. And Daniela will present a talk entitled Rhythmicity of Foraging Activity in Leaf-Cutting Ants, the Influence of Molecular and Environmental Factors. So nice to meet you, Daniela.
Hello.
I'll let you share your screen.
Oh, do I already start now?
Yeah, you can start, yes.
Okay. Hold on a second. Let me share my screen. Okay. Can you see this?
Not. Yes, I
Yes.
can see. Yes, it's full screen now. Perfect. Have a nice talk.
Okay, thank you. So do I start now?
Yes, you can start.
Okay, thank you. So first, let me thank the organizers of this very wonderful symposium and for giving me the opportunity to speak. My talk will be about rhythmicity of foraging activity in leaf-cutting ants, influence of molecular and environmental factors. Social insects, so that's bees, wasps, termites, and ants, go out of their nest to forage. They forage large quantities of food, not for themselves, but they are central place foragers. And that means they bring these foraged material back into the nest to be distributed among the colony members. What we see in the field is actually that these colonies do not forage all throughout the day and night, but we see rhythmicity of their foraging behavior. Here, for example, we have two ant species, Camponotus mus and Camponotus rufipes, and they occur in the same habitat and avoid competition with each other by one species foraging during the day and the other species foraging during the night. So it is advantageous to time your foraging behavior. You even increase the chances to discover food, and you can avoid unsuitable or even harmful environmental conditions. This is what this leaf-cutting ant Adaptations colony did. First, they were most active during the day. Here you can see this, the peaks of height of activity were around 12 o'clock at noon. And then 2 months later, they had actually switched to night foraging, and the high peak was around midnight. To be able to time behavior, to tell time, insects and other animals have evolved an endogenous clock. And this endogenous clock consists of an input pathway, the central oscillator, and an output pathway. And in insects, the endogenous clock is situated in the optic lobes of the insect brain. This clock has a so-called free running rhythm of approximately 24 hours. That is why it's called a circadian rhythm. The clock needs to be set and continuously reset by external Zeitgebers. Sorry for the German word, hard to pronounce. What it means is time giver cues. And the most prominent ones in the environment are either the light-dark cycle or temperatures, because it also fluctuates throughout the day and also throughout the year. There's also a gene associated with foraging. It's aptly named the foraging gene. It was first discovered in Drosophila melanogaster fruit flies, and in recent years it has become apparent that this gene is not just associated with foraging, but with division of labor in general. And division of labor in social insects is also associated with age. That means that young workers stay in the nest to perform in-nest tasks such as brood care, while the older workers leave the nest and go out to forage. What we know about foraging gene expression in social insects is that in Apis mellifera, the honeybee, the foraging gene expression increases with age. So foragers have the highest level of this foraging gene expression, while in the seed harvester ant Acromyrmex barbatus, it's actually the other way around. And here, younger workers have the highest foraging gene expression levels. And in a more recent study, it was also shown that foragers itself have fluctuating foraging gene expression levels, and they have the highest levels when they are actively foraging and lower levels when they are inactive. Now, leaf-cutting ants have the largest colony in all social insects. They have or can have millions of individuals, and they create these large underground nests. that all speaks to their high ecological success. And that is actually based on their evolution of being able to farm a symbiotic fungus. And that is why they need the large amounts of fresh plant material you see them carrying into the nest. It's not to sustain themselves, but to actually grow this symbiotic fungus. And that is their food source. What we are interested in our work group is to discover the mechanisms that are on the basis for the rhythmicity of foraging we see in these ants. So for this study, we had 3 research questions. First, do abiotic factors influence the daily pattern of foraging activity of leaf-cutting ant colonies? Second, are the displayed activity rhythms driven by an endogenous clock of the ants? And is there a connection between age and with it division of labor and foraging gene expression in leaf-cutting ants? So first, we quantified colony foraging rhythms of a whole colony in a climate chamber. And what we did is we put a colony of Acromyrmex lundii in here in this closed box. So all the fungus chambers were under constant darkness and a nice warm 25 degrees to simulate underground nest conditions. And the workers could leave the colony and reach this foraging arena where we offered leaves ad libitum throughout the day and night. And we put a camera across the foraging trail and filmed continuously during this time and could so quantify the number of outgoing and ingoing laden and unladen ants. We performed this experiment under 2 so-called seasons. We first simulated the spring season with a cold night at 10 degrees Celsius and a nice warm day at 25 degrees Celsius. And under summer conditions, we had a nice warm night, 25 degrees, and a hot day at 35 degrees Celsius. To also simulate the solar radiation during this time, we put a heat lamp over parts of the path. And we let this experiment under these environmental conditions run for 14 days each. The first week was to get the colony acclimatized to this condition. And then the last week of these 2 weeks, we used to analyze foraging activity of a whole colony. At the end of the 2 weeks, we then collected foragers, laden foragers on the way back to the nest. And then we split these into 2 groups. And in the first group, we wanted to determine the activity rhythms now of solitary foragers in comparison to in the colony context. And the other group we used for foraging gene expression levels using qPCR. What we also did and wanted to know what were the activity rhythms of age-known workers. Foragers are usually older workers, but we don't know how old they really are. So we created age-known workers by raising them from the larval stage, and so created 0-day-old workers right after eclosion, 7-day-old workers that are young workers, usually performing in-nest tasks, and 21-day-old workers, and these are old enough to leave the nest to forage. And of these age-known workers, we also quantified the foraging gene expression levels. Here, this is a picture of 2 of the activity monitors we used for the solitary workers. In each of these plastic tubes, there was 1 ant that could walk back and forth in these tubes. And on each end, we put a little food, so they had enough food to sustain themselves. We left these ants in here for 7 days. And during these 7 days, the ants encountered stable environmental condition in the form of 25 degrees Celsius, so now it's in warm, and a high relative humidity. But one group encountered an external Zeitgeber cue of the light-dark cycle, so 12 hours light, 12 hours dark, and the other group we kept under constant darkness, so here the rhythms we see are solely based on their entrained endogenous clock. And when these workers were walking back and forth in this week, they broke or walked through a light beam, and this was automatically counted. And what you then get at the end of these 7 days is a so-called Actogram if you plot these beam crosses. And here, as an example, I've shown you one from a Camponotus rufipes worker. Here, they were kept under constant darkness, so it was also dark here in the gray color during the normally light day, and of course, dark during the night. And as you can see here, this sole worker had a very, very precise rhythm, even under constant darkness for 21 days in this activity monitor. So a very precise rhythm by the endogenous clock. What we used for analysis was the program called Actogram J. That's a plugin of ImageJ, so you can download it freely from the internet. If anybody is interested in this type of research, you can download it. Just look for Actogram J. And what this program allows you is to analyze with a click of a button certain variables. And we chose to look at periodicity. So what is the rhythm of the end? How strong was this rhythm? And when were these animals most active? Then for the qPCR analysis, we flash froze the whole worker, removed the head, then removed the brain, and then used this brain tissue for our qPCR analysis. And what we got from this qPCR analysis was the relative foraging gene expression levels of these animals. So on to the results. First, the colony foraging activity during the spring and the summer. Here on the upper part, this is the spring. You see the temperature curves with a nice warm temperature during the light phase and the cold temperature during the dark phase. And what you clearly see is here, this colony of Acromyrmex lundii was day active and not night active. And this changed during the summer conditions. Here we have now the hot temperatures during the light phase and the nice warm temperatures during the dark phase. And here the colony had switched to nighttime foraging activity. But what about the activity of solitary foragers we took out of this group context? First, the periodicity, that is the rhythm of these ants. Here with a red dashed line, I've marked this is a precise 24-hour rhythm. That's 1,440 minutes. And as you can see, with the influence of an external time giver cue, a zeit giver cue of the light-dark cycle, these animals, these foragers, also show a very precise rhythm of 24 hours. When this zeit giver cue is missing, as I said, the endogenous clock is free running of approximately 24 hours. And here leaf-cutting ants seem to be having a longer than 24-hour free running rhythm of the endogenous clock. So how strong was now this rhythm of these ants? It's called the power of the periodicity. And with the light-dark cycle, with the external zeitgeber cue, again, the power was higher of this rhythm than without this zeitgeber cue was missing, and the animals simply relied on their endogenous clock. When was now the height of activity of these animals? Height of activity is also called the acrophage. Here on the y-axis is time of day. And I want you to remember that 6 o'clock is the lights on, the beginning of the light phase. And here we can see these animals as foragers from the spring season. They had a height of activity at 12 o'clock, that is right in the middle of the light phase. And when the zeitgeber cue was missing, then the height of activity was slightly pushed to further along the day, but still during the day, the light phase. So these animals were still most active during the day. And this changed now when we look at the summer foragers. As you remember, the colony was night active, but now the height of activity of these solitary foragers was again at 12 o'clock, right in the middle of the light phase. So these animals had actually switched from nighttime activity in the colony setup to daytime activity in the monitors. What about now the activity of these age-known ants? Unfortunately, I cannot show you any activity from the 0 day, from the freshly closed workers, because there was a really high mortality in the monitor, so I could not do any statistics. So I will only present you the results from the 7-day-old workers. Remember, these are in-nest workers, and the 21-day-old workers. These are older workers that leave the nest to forage. So what we can see here, with a zeitgeber cue of the light-dark cycle, there is no difference in rhythmicity between young workers and older workers. They have both a very precise rhythm of almost, or here even identical, 24-hour rhythm. And to our surprise, what we saw also when this zeitgeber cue is missing, then they still have a very precise rhythm, even very, very young workers, only 7 days old. Now, the power of this rhythm only slightly increased with age, and only here in the setup without an external time giver Q. Now, the qPCR analyzes foraging gene expression levels slightly increased with age. And when we compare this with the age unknown foragers, which we think are older, there's still no difference in in the foraging gene expression levels, unlike we saw for other social insects. So to conclude, the high temperatures lead to a switch to nighttime foraging in leaf cutting ants. And we think this is not due to a preference for actually foraging under darkness, but rather based on avoidance of high temperatures. We think this is based on the results from the monitor experiments, where these formerly night active foragers from the summer setup actually switched back to daytime activity as soon as the temperature during the day was also nice and warm and no longer too hot for them to be outside. Then leaf cutting ants seem to have a very weak entrainment of the of the endogenous clock, and they rather react to local zeitgeber cues, that is light and temperature. And there is no difference in foraging gene expression levels between young workers and older foragers, unlike we've seen in other social insects. So as a take-home message, leaf cutting ant foraging behavior seems to be guided by local environmental cues at the moment, rather than on internal clock, rather than based on the internal clock or on foraging gene expression. Last but not least, I would like to thank Anna Soltani. All what I presented here today was from her bachelor thesis. She did a tremendous amount of work. I would also like the work group of Markus Tam for help with the molecular part of this study, and my supervisor, Flavio Rosses. And of course you, because this is the last talk of the morning session. You're all with me, so thank you for your attention, and I'll be happy to take questions.
Thank you very much for your talk, Daniela. Very impressive empirical work. Very interesting. I'm still waiting for questions in the chat. So I can maybe start with a few short questions.
Okay.
Maybe a very small technical one to start. I didn't really get what was, let me look at my notes, the power of periodicity measure you had. I didn't get this one. Could you maybe just...
Yes. The power is, that is a dimensionless number. It just shows you how high and relative to the other power was the strength of the period. So the higher the number, the stronger the animal stuck, let's say, to their daily rhythm.
Okay, okay. And maybe another easy question to ask, but difficult to answer. Do you think like this set of experiments is maybe interesting to study, to maybe start to see how global warming could affect the foraging of ants? Because you have some evidence that temperature has quite a lot of effect on the foraging behavior, right?
Yes, yes, definitely. I think you can definitely use this setup. It would be nice to compare different animals, maybe even species, from invasive species that we think that the height of their ecological success in these areas where they're invasive might be because they are better adapted to global warming. So you can definitely test what their foraging activity or their internal rhythm is compared to other ants, if this is the basis, because that they are so invasive and successful.
Yeah, and I think it's very interesting how your setup allows to compare the individual behavior with the colony choices.
Exactly.
We've got some questions in the chat.
Perfect.
So first question from Elise Jan. Thank you for this very interesting talk. Is there no impact of colony isolation on solitary workers' behavior?
Yes, yes, definitely, especially for leaf cutting ants. I showed you before, let's see, let me switch back. This is, as an example, this Actogram from Camponotus rufipes. Then they survived. So each line here is a day in the monitor. And they survived for at least 21 days in these monitors. And for leaf cutting ants, I could only test 8 days. They didn't even last 14 days because I had planned to first test them, the same foragers under light dark conditions, and then the same forager under complete darkness for another week. And they didn't survive for 14 days. So there's definitely differences, species differences. And I think leaf cutting ants are so social that they are not able to survive for longer periods on their own.
Okay, thank you. Second question from Erika Dawson. Hi, Daniela, nice talk. Is it known if the ants undergo any sensory adaptations when they switch to nighttime foraging?
Good question. I must admit, I'm not able to answer this question. So what exactly is meant by this? What kind of adaptation?
So I can repeat the question.
Yes.
I think specifically it's sensory adaptations.
Oh, okay. Yeah. Again, leaf cutting ants, they have not the best eyes. They can certainly see, but even during the night, they have very successful foraging bouts. So they are very, very active also during the night when you see them switching to nighttime activity. And that is, they have this really developed trail system. So they have clear foraging trails that they also mark with pheromones. So they're not really reliable or relying on optic orientation. So no, I don't think there's actually a decrease of foraging during the night. So that should not have any effect on nighttime foraging.
Yeah. Another question from Charlie Zoyo in the chat. Thanks, great talk. Is a single environmental cue, only light or only temperature cycles, enough, or both of them are needed to affect the foraging activity?
What we saw in these monitor experiments, because we kept the temperature stable and the humidity stable and only changed the light conditions. So this was enough for the ants to orient themselves and to show a rhythm. The same, I would think, with temperature. We didn't test this, but we tested this in other ants. And there, again, there was with Camponotus ants, what was found that light is a stronger Zeitgeber cue than temperature, but temperature itself also works, but to a lesser degree.
Okay. And maybe a last question before closing this session up. Question from Said. Thanks. Would that be also to explore anthropogenic light at night? Elaine? Oh, sorry, I don't understand what that means, Said. So let me start again. Would that be also to explore anthropogenic light at night? Elaine, impacts of ants under laboratory conditions? And what would be your expectations in terms of biological relevance in captivity? Did you get that?
Yeah, I think I know what he means.
Okay.
These animals, we also keep them in a climate chamber, as they are usually ants from the tropics, and there they are continuously kept under light-dark cycle of 12 hours light on and 12 hours light off. So I don't know how highly entrained the animals are already before we used them for the experiments, and that's why we gave them 1 week when we put them in this test chamber to acclimatize to our conditions. But we also used the 12-hour lights on, lights off light cycle. I have a cooperation with a lab in Uruguay at the University of Montevideo, and they actually test field workers or field animals collected from the field in these activity monitors under the adjusted light-dark cycle that they also would see in nature. And the results are certainly a bit different. But what they also found is that light is a very strong Zeitgeber cue, whether the animals are from a captive from a lab or directly collected from the field.
Okay, thank you very much, Daniela. I think maybe if people have other questions, I can ask in the Discord channel dedicated for your talk. And thank you again for your very nice talk. And thank you to all our speakers this morning It was very nice having you all here. Now there is a break, and an hour-long break, and then we've got some social events at 11 GMT, so in 1 hour. I think, yes, I see the link in the chat, and the first of these social events will be a Meet the PI mentoring session with one of our plenary speakers, Anna Silva. And then we will have a meeting with a social media expert, Dr. Catherine Scott. And that's it for this morning. Thank you all again, and I see you around. Bye-bye.